two pathways between fatty acid synthesis and between beta oxidations. There is several differences between these two processes. The first of all, fatty acid biosynthesis, I said, takes place in the cytoplasm. In contrast, the beta oxidation primarily happened or occurs in the mitochondrial matrix. The first difference. The second one, the intermediate in the fatty acid biosynthesis are covalently linked to what is called sulfhydryl group of acyl carrier protein. This is abbreviated as ACP, whereas the intermediate in beta oxidation are covalently attached to sulfhydryl group of coenzyme A, اللي هي acetyl coenzyme A. Each product in the beta oxidations, the end of the product called, for example, beta ketoacyl coenzyme A. This is the third step of beta oxidation. The first step starting by activated acyl coenzyme A. So the intermediate is covalently attached or binding to the sulfhydryl group of coenzyme A in beta oxidation, while in fatty acid synthesis attached or covalently linked to sulfhydryl group of acyl carrier protein. This is the second difference. The third difference, the enzymes of fatty acid biosynthesis in higher organisms like a human being are joined in single polypeptide chains يسمونه fatty acid synthase fed polypeptide بها 7 enzymes. These 7 enzymes connected together to form fatty acid synthase complex enzyme. شو يسمونه fatty acid synthase complex enzyme. ليش سموها complex? Because it's contain 7 enzyme connecting together or joined together. In contrast, in beta oxidation, each enzyme acts isolatedly, alone. For example, as hydratase enzyme or acyl coenzyme A dehydrogenase enzyme or beta ketoacyl coenzyme dehydrogenase enzyme and thiolase enzyme, as we said in the previous lectures. The fourth difference you have to know in the تكوين الفاتي acid, the growing fatty acid chain is elongated by the several steps, by the sequential addition of 2 carbon atoms, just like the removal of 2 carbon atom in beta oxidations. منين تجينا هاي 2 carbon atoms؟ تجينا طبعا منين؟ derived from the acetyl coenzyme A. While there in beta oxidation, removal of 2 carbon atoms in form of acetyl coenzyme A. But here, in fatty acid synthesis, the activated donor of 2 carbon atoms in the elongation step نسميه malonyl acyl carbon protein, acyl carrier protein. malonyl acyl carrier protein. شنو هذا المالونيل؟ شنو اختلاف عن الاستايل كوانزيم A؟ الاستايل كوانزيم A طلاب هو 2 carbon atom compound, while the malonyl acyl carrier protein contain 3 carbon atoms. So give 2 carbon atoms to elongate and 1 carbon atom is removed in form of carbon dioxide. So the elongation reaction is driven by the release of carbon dioxide. Please, is that clear? If جدول inshallah clear. The fifth difference, the reductant in the fatty acid biosynthesis is not pH, whereas the oxidant in the beta oxidation are 2 type, والناد والفاد, this oxidant, while here in the fatty acid biosynthesis هو الreductant يسمونه not pH. The last difference, the elongation by the fatty acid synthase complex will stop at the formation of the palmitic acid, يعني carbon number 16 ويتوقف formation of fatty acid biosynthesis. The further elongation, further addition of carbon atoms to form long and very long fatty acid occur in microsomes, and we're talking later about this process. So further elongation and insertion also of double bonds are carried by other specific enzymes. This is That's all about the difference between fatty acid synthesis and between fatty acid degradation, I mean beta oxidation. I will talking briefly about de novo biosynthesis of fatty acid lipogenesis, which is occur in the cytoplasm. Please, you will see details here. The details, this is for you to understand what goes on here. But what is requested from you, what I say, this important, the major things, this all essential to know about the fatty acid, you have to know by each one of you. So a large proportion or a large portion of fatty acid used by our body supply from diet. Not all the fatty acid is synthesized by de novo fatty acid biosynthesis. Diet fatty acid. طب شلون يجينا من الدايت الفاتي acid in form of fat. Okay, come in form of fat in form of triglyceride بالكيلومايكرون ويدخل لنا الليمفاتيك سيركوليشن وبعدين يدخل لنا البلد سيركوليشن. لكن إذا if there is a plenty of carbohydrates or proteins and other molecules obtained from our daily diet consuming in excess of body needs زائد عن حاجة الجسم, هاي الكربوهيدرات والبروتينز can be converted to fatty acid to be stored as a adipose tissue in form of triacylglycerol اللي هو triglyceride. هاي لازم تعرفها طلاب. هذا naturally occurring in our body. So this system present in many tissue including liver, kidney, brain, lung and mammary gland and adipose tissue. As you know, the major things should be available for beta oxidation in the activation step and during beta oxidations, ATP, NAD and fat. Here, the most important requirements including NAD pH, ATP, manganese, biotin, and bicarbonate, which is so important because it is a source of carbon dioxide. What I want to know, the acetyl coenzyme A is starting molecule, which is called immediate substrate, while the palmitate, which is the end product, اللي هو fatty acid, newly fatty acid product. As I said, there is NAD pH as a reducing agent. From where does NAD pH come? So the question here, or the issue here, what are the sources of NAD pH for the fatty acid biosynthesis? This is an important thing. Of course, you know, in the carbohydrate metabolism, as you know, glycolysis, gluconeogenesis, glycogenolysis, and glycogenesis, there is what is called a pentophosphate pathway in carbohydrate metabolism. And this pentose phosphate pathway occur in the cytoplasm, and the fatty acid biosynthesis occur in the cytoplasm. Okay? So, the pentose phosphate pathway is the major source of NAD pH. And there is no barrier between these two processes, as they are both of them occur in the cytoplasm. So the NAD pH come from pentose phosphate pathway to be incorporated or involved in fatty acid biosynthesis. This first and the major source of NAD pH. And the second common source of NAD pH, which is called malic enzyme, which is an enzyme catalyze the conversion of malate to pyruvate in the cytoplasm after exiting of malate from the mitochondrial matrix to the cytoplasm. And the third source, which is not present in human, which is called extramitochondrial isocitrate dehydrogenase reactions, which is another source of NAD pH. As I said, the fatty acid biosynthesis occur in the cytoplasm, and the immediate substrate and the building block of fatty acid synthesis is acetyl coenzyme A. And the acetyl coenzyme A is present inside the mitochondria. So by which mechanism the acetyl coenzyme A getting out of mitochondria to be available in the cytoplasm to be ready to involve in fatty acid biosynthesis. So there is an important shuttle as you see in this figure, and this is requested by each one of you. This is a citric acid cycle. Please focus with me. Pay little attention. As I said, the glycolysis ending, as is glucose, ending to form a pyruvate. And the pyruvate, by the pyruvate dehydrogenase, will convert it to the acetyl coenzyme A. Just like the conversion of, regarding protein catabolism, or amino acid catabolism, will form acetyl coenzyme A. So the acetyl coenzyme A, what from amino acid catabolism or protein catabolism, will form acetyl coenzyme A. Of course, the protein catabolism or amino acid catabolism occur inside the mitochondria, while the glycolysis in the cytoplasm, but pyruvate can enter freely the mitochondrial membrane, and by the pyruvate dehydrogenase will convert the pyruvate to acetyl coenzyme A. At this step, we will form acetyl coenzyme A. As you see, the intermediate of citric acid cycle, starting by citrate, oxaloacetate, malate, oxaloacetate, and etc. And this cycle repeats itself more and more. Acetyl coenzyme A will be binded to oxaloacetate to form citrate. This reaction catalyzed by citrate synthase enzyme. This is inside the mitochondria. When we form citrate, there is in the outer mitochondrial membrane citrate shuttle or citrate channel. This is specific for citrate, and the citrate can easily move out the mitochondria to enter the cytoplasm. Inside the cytoplasm, the citrate again dissociated again to its precursor اللي هم شنو هو الاوكزالواسيتيت والاسيتايل coenzyme A. By which enzyme? By enzyme called citrate lyase. Citrate lyase will catalyze the dissociation of citrate to oxaloacetate and acetyl coenzyme A. So the acetyl coenzyme A ready to be involved in fatty acid synthesis. What about the oxaloacetate? راح نستفاد من الاوكزالواسيتيت كلش. شلون نستفاد منه؟ احنا نريد a source of NAD pH. The oxaloacetate again by malate dehydrogenase convert the oxaloacetate to malate. So we have two fate للمالate. Either directly enter the mitochondrial matrix or to be involved in citric acid cycle, or this malate converted to pyruvate by malic enzyme, and this will give us NAD pH, which is the second common source of NAD pH that's required for fatty acid synthesis. And pyruvate can easily enter the mitochondrial membrane to be involved in citric acid cycle. Is that clear? Clear? Answer me. Clear?
inshallah دكتور.
Ya Allah, inshallah clear. So we'll shift to another topics. راح نبدي شلون دا تصير عملية الفاتي أسيد بايوسينثيسيس. قلنا الفاتي أسيد هو الإميديت سبستريت هو شنو؟ الأسيتايل كوإنزيم A. والدونر of two carbon atoms that's added to acetyl coenzyme A in each turn هو المانونيل coenzyme A. ابتداء هو المالونيل coenzyme A يتكون منين؟ من الاسيتايل coenzyme A والبايكربونات ATP. هو ضفنا له فد كربون لأن الاسيتايل coenzyme A هو two carbon atoms to be form malonyl coenzyme A should added another carbon which is come from bicarbonate. So the fatty acid biosynthesis start with carboxylation of acetyl coenzyme A, and this is irreversible reaction called the rate limiting step or committed step in fatty acid biosynthesis. And in every metabolic process, there is a committed or rate limiting step. Here, the rate limiting step is the conversion of acetyl coenzyme A to the malonyl coenzyme A, and this reaction is catalyzed by important enzyme called acetyl coenzyme A carboxylase enzyme. Of course, this reaction اللي هو الأسيتايل coenzyme A plus ATP plus bicarbonate of two-step reaction, not one-step reaction. That's for you. You should know this reaction is two-step reaction, not one-step reaction to form of malonyl coenzyme A. After the formation of malonyl coenzyme A, the synthesis, as I said, the synthesis or synthesis of malonyl coenzyme is catalyzed by acetyl coenzyme carboxylase enzymes اللي هو has requirement of vitamin biotin and others, as I said, the manganese and bicarbonate. هنا يجي دور المالتي انزيم بروتين اللي هو شنو هو الفاتي اسيد شنو synthase complex enzyme اللي هو contain 7 enzymes. طبعا قلنا هذا فد دايمر يعني فد polypeptide has two identical subunits. Each subunit has 7 enzymes. These subunits or polypeptide connecting or arranged in tail or head tail or tail head arrangement. Okay, as you will see in the next figure, this is the two-step reaction of formation of malonyl coenzyme A. The first thing, carboxylation of biotin involving ATP. Biotin enzyme plus ATP bicarbonate. This will form carbon dioxide biotin enzyme plus ADP and organic phosphate. The second step of this rate limiting reactions هو the conversion or the carboxylation of acetyl coenzyme A by carbon dioxide biotin enzymes to form the malonyl coenzyme A plus biotin enzyme. So the biotin reused to another reactions. So the bicarbonate is the source of carbon dioxide here in these reactions. As I said, this is an rate limiting reaction. So any rate limiting reactions play a key role in the regulation of this metabolic process. So by which mechanism هو الأسيتايل coenzyme A carboxylase هو المنظم الريت limiting step بالفاتي acid synthesis. The acetyl coenzyme A carboxylase itself had been regulated. By which mechanism we can regulate the activity of acetyl coenzyme A carboxylase enzyme? We have two types of regulation here. The first one called short-term regulation of acetyl coenzyme A carboxylase, and the second called long-term regulation of acetyl coenzyme carboxylase. We start to discuss the short-term regulation of acetyl coenzyme carboxylase. Of course, this enzyme naturally present or available in active form. Okay? شلون active form؟ هو دايمر بروتومير دايمر اللي هو acetyl coenzyme A. This enzyme undergoes allosteric activation. هذا الاكتيفيشن طبعا keep in your mind، حفظوها عني طلاب. معظم الريت limiting enzymes معظمهن هو يتحفز activated by the first substrate of another metabolic process and inhibited by the end product of the same metabolic process. شلون يعني؟ الاسيتايل هو كاربوكسيليز، اسيتايل كوانزيم اي كاربوكسيليز انزيم، قلنا هو ان اكتيف فورم، هو دايمر. شلون يصير بي اكتيفيشن؟ الستريك اكتيفيشن. ستريت من يطلع من الميتوكوندريا للسيتوبلازم. once the citrates become in the cytoplasm أصبح شنو؟ أصبح ready to be involved in the fatty acid biosynthesis. So the enzyme will be activated. شلون يصير بي activation؟ يصير بي polymerization. لذلك يصير active form acetyl coenzyme A carboxylase enzyme. شلون يصير بي inactivation؟ by the end product of the same metabolic process هي الفاتي acid biosynthesis. شو تسوي؟ شو تخلق لنا؟ غير تخلق لنا فاتي acid. إذا صار لنا mature fatty acid for example palmitic acid 16 carbon atom fatty acid. once this achieved عندما يتكون ال 16 carbon atom fatty acid this will allosterically inhibit the acetyl coenzyme A carboxylase. This is one form of short-term regulation of acetyl coenzyme carboxylase enzyme. Okay? هاي وحدة. The second mechanism of short-term regulation is achieved or accomplished by reversible phosphorylation. You should know always, almost, usually the phosphorylation will convert the enzyme from the inactive form to active form with a specific or some exception. Here, this enzyme, one of these exceptions. What I mean by that? In the presence of counteracting regulatory hormone اللي هم glucagon and adrenaline or epinephrine. الابنفرين and glucagon شو يسوين؟ يسوين phosphorylation للاسيتايل coenzyme A carboxylase. Once the phosphorylation achieved the enzyme become inactive. طبعا هاي وين تصير؟ طبعا هذا common sense. شلون common sense؟ common sense لان احنا اذا ذن الهرمونات الكاونتر اكتنج هرمون ايش وقت come to play؟ ايش وقت تطب الساعة على مود تشتغل؟ بالفاستنج and starvation state. زين الفاتي اسيد بايوسينثيسيس يمتى تصير؟
بالفيد ستيت.
تلقى الانسولين يشتغل. اني كاتابوليك بروسيس تلقى الكاونتر اكتنج هرمونز يشتغلن. keep in your mind this information please. So in fasting state adrenaline and glucagon will enhance the phosphorylation and the acetyl coenzyme A carboxylase enzyme will be inhibited. خلصت الشغله. in feeding state الانسولين come to plays or to play sorry. so شو يسوي لنا الانسولين؟ يسوي لنا dephosphorylation of acetyl coenzyme A carboxylase. once the dephosphorylation achieved the carboxylase enzyme become active and the fatty acid biosynthesis will go ahead or go on. Okay, this is called second mechanism of short-term regulation of acetyl coenzyme A carboxylase enzyme. Regarding the long-term regulation of acetyl coenzyme A carboxylase enzyme كلش easy. شلون يصير long-term regulation؟ إذا عندنا if there is a prolonged consumption of a diet contain plenty or excess calories such as high carbohydrate diet. As I said, if there is a plenty of carbohydrate and protein that exceed the ability of our body. This will convert to be used as a glucose. This or as said, okay, right, glucose, because the alanine can converted to pyruvate and form in gluconeogenesis to glucose. This will convert these excess carbohydrate to lipid, and this enhance the fatty acid biosynthesis. Conversely, low caloric diets in fasting state causing a reduction in fatty acid biosynthesis. ليش يصير by decreasing the synthesis of، يعني شنو decreasing synthesis ليش؟ يعني الاسيتايل كوانزيم كاربوكسيليز طبعا إلى synthesis. شو وقت يزداد السينثيسيس of acetyl coenzyme A carboxylase؟ هاي يسمونه increase the gene expression. Once the gene expression increased, the acetyl coenzyme carboxylase synthesis increased. يمتى يصير increasing بالجين expression of this enzyme occur in شنو in fed state, while in fasting state will be decreased. So this is a clear, so clear. And this figure shows the process of regulation of acetyl coenzyme. As you said, the citrate will trigger the acetyl coenzyme activate. I mean, this is in a green triangle or in green circle. This will trigger and stimulate the acetyl coenzyme A carboxylase. In addition, the insulin in feed state will activate the acetyl coenzyme carboxylase by dephosphorylation, while the malonyl coenzyme A, as it is an intermediate product of fatty acid biosynthesis, will form ultimately the palmitoyl carbon 16 carbon atom fatty acid. This the end product will inhibit the acetyl coenzyme A carboxylase, and the counteracting hormones like glucagon and adrenaline, which is acting in fast state, will inhibit again the acetyl coenzyme A carboxylase. This is regarding the short term and long term regulation, or only short term regulation. This figure, anyone answer me?
Short term.
Only short.
Short term.
Excellent. This is regarding the short term regulation, because the long term depend on the much or plenty or excess amount of calories, like in carbohydrate rich diet. This is regarding the fatty acid synthase complex. This is for observation. You have to know, please, this 7 enzyme bind or linked or joined together in what is called head tail arrangements. As you see in blue, the thioesterase enzyme on the right hand, while the thioesterase in the white color from the left hand. Both of these 7 enzymes or polypeptide chain or monomer or subunit, each subunit from each end connected. This is requested, only this one requested. By which mechanism connected? As we said in the differences between the intermediate of both enzyme connected there in coenzyme A, and here in sulfhydryl of شنو? Of acyl carrier protein. Sulfhydryl of cysteine in the ketoacyl synthase will be connected to the sulfhydryl of 4-phosphopentathene of acyl carrier protein. Only this I want you to know, and this is an important to know, because sometime I will ask you about this. Of course, the fatty acid synthesis, as I said, is just a reversal process of beta oxidation. What I mean? I mean there is a 4 steps of fatty acid synthesis, and this only for observation. This the fourth step of fatty acid synthesis. Only this requested from you because there is a details you will see in the end of the lecture, details about each step. I want you to know only these four steps. The first step is as we said, the condensation of activated acyl group in form of acetyl coenzyme A and two carbon derived from the malonyl coenzyme A, which is formed by carboxylase enzyme to form a 4 carbon atoms compound, and this will extend the acyl chain by 2 carbon. The mechanism of the first step of this reaction is given. حكينا illustrated the role of decarboxylation in facilitating the condensation. قلنا هو المالونيل 3 carbon atoms والاستايل coenzyme A 2 carbon atoms when bind or join together there is a decarboxylation process and release carbon dioxide in form of carbon dioxide and will form 4 carbon atoms. The second step هو البيتا keto group is reduced من أقول reduce يعني شنو الإنزيم هناك إذا صرنا أوكسيديشن قلنا دي هيدروجينيز إنزيم، لا هنا another enzyme راح نحكيه هنا واحد and the third step
reductase enzyme مو دكتور؟
excellent. And the third process, the elimination of water will create double bond, while in beta oxidation ايش قلنا هنا هو elimination. فإذا elimination هناك hydratase هنا شو يسمونه dehydratase. And the fourth steps, the double bond is reduced. That means breakdown to form the corresponding saturated fatty acids. الحكي هذا حقيقة أريد تعرفوا تقروا على مود تفتهم what's going on here in these four steps, and all the four steps, and I discuss them in details here. This is all for observation, even this, but here what I want to see condensation process. شنو ال rate limiting here enzyme, with which enzyme catalyze this process. قلنا الأسيتايل coenzyme A, carboxylase enzyme. The second steps by which enzyme as أحد طلاب هسه جاوبني الله يخليه هو الكيتو أسيل كيرير بروتين ريدكتيز انزيم. والديهايدريشن هو عكس البيتا اوكسيديشن، هناك هيدراتيز هنا دي هيدراتيز enzyme. The last one step 4 هو reduction of the double bond by enol acyl carrier protein reductase enzyme. The name of enzyme, please, you have to know name of enzyme for each 4 step, the name of the step, and each enzyme catalyze this step مطلوبة من عندك يعني. Okay, and these 4 steps will repeat again and again to form the final requested product اللي هو carbon 16 palmitic or palmitol acid. Our body ability يكون لنا 16 كربون بالسيتوبلازم. زين إذا احنا نريد فد long and very long chain fatty acid شلون راح نسوي؟ There is an additional mechanism in our body or pathways called microsomal system, which is responsible for elongation of saturated and unsaturated fatty acid and for insertion again of double bonds. Okay, هذا السيستم starting from the carbon 10 احنا خلقنا الكربون 16 زين by adding 2 carbon atoms using malonyl coenzyme A as acetyl donor and not BH as a reductant. The same thing for the process of de novo biosynthesis of fatty acid and catalyzed by microsomal fatty acid elongase enzyme. بس اسم الانزيم يا اخوان ما يعني هو ما بيشرح ولا بيه حكي هواي زايد. Only fatty acid elongase responsible for the elongation of fatty acid that's formed by de novo fatty acid biosynthesis. When occur, the site of occurrence, the site of this elongation when they يصير بالميكروزومال system. هو مثل ما تشوفون يسوي لنا كربون 22 24 acid for sphingolipid. وقلنا هو البالميتيت is the precursor of other long. هو البالميتيت اللي كوناه بالدينوفو fatty acid biosynthesis هو ال precursor of elongation process that occur in microsomal system. اوكي. elongation system وين موجود؟ قلنا هو الميكروسومال. وين الميكروسومال سيستم؟ بالاندو smooth endoplasmic reticulum and in mitochondria also. And this is a process of elongation and desaturation. Okay, the process of elongation and desaturation only for observation and only knowing the name of the enzyme and the site of occurrence. Of course, the desaturation process in our body, you have to know these facts. Palmitate and stearate serve as precursor of the two most common monounsaturated. نقول desaturation كل الحكي مالتنا كان عليه عن saturated fatty acid. زين شلون نخلق unsaturated fatty acid؟ by desaturation system. The two most common monounsaturated fatty acid of animal tissue اللي هي البالميتوليل or palmitoleic acid اللي هو 16 when that's mean contain single double bond between carbon 9 and carbon 10 and oleate اللي هو 18 carbon atom with single double bond. The same thing between the carbon 9 and 10. Both of these have a single cis double. وقلنا most commonly naturally occurring شنو؟ unsaturated fatty acid بالcis configuration. اكو trans اكو بس ال commonly or most common or naturally occurring هو in cis configuration between carbon 9 and carbon 10. اللي لازم تعرفوا the double bond is introduced into the fatty acid chain by an oxidative reaction catalyzed by fatty acid coenzyme A desaturase enzyme. من اسمه يعني ما عندك دوخة بيه. هو desaturase enzyme in bracket هو mixed function oxidase. بالمامليان يعني المامليان including human being hepatocyte can readily introduce نستطيع insertion of double bond only at شنو delta 9 position of fatty acid but cannot introduce additional double bond between carbon 10 and the methyl terminal end. هاي هم لازم تعرفوها. So thus mammals cannot synthesize lonylate 18 with 2 double bond or alpha lonylate with 3 double bond. فقط هاي as information true false MCQ you should know. And this is a process of desaturation وأن حقيقة فقط for observation. Regarding the medical importance كلكم تعرفون مثل ما قلنا هسه إذا واحد ياكل fat or carbohydrate rich diet and fat rich diet protein rich diet واحد الله يسلمه ياكل هواي شنو الإند؟ الأوبيزيتي مو تمام لو لا become obese راح يسمن due to شنو deposition of fatty acid in adipose tissue whether in visceral adipose tissue طبعا داخله بالفيزرال الأعضاء الداخلية الشحم داير ما داير كلياته داير ما داير الكبد مالته داير ما داير الأومنتم والميزنتري هذا يسمونه visceral fats or subcutaneous fat. لذلك increase the body weight beyond the 40 بالميل يسمونه obesity وبالفيميل beyond 35 inch إذا هذا السيركمفيرنس مالتها قسناه هذا شي نسمونه obesity. To prevent this medical problems we have synthesized an artificial drugs called hydroxycitrate. شنو هذا الهيدروكسيستريت؟ هذا الدراجز اللي هو ATP ستريت لايز تتذكرون بالسيتوبلازم. هسه من حكينا قبل شوية، طلعنا شوية. Remember what I said about the movement of the citrate from the mitochondria to the cytoplasm. In the cytoplasm citrate lyase enzyme will dissociate the شنو citrate to oxaloacetate بعد، والاسيتايل coenzyme A على مود ناخذ الاسيتايل coenzyme A to be involved in fatty acid biosynthesis. احنا ما نريد الاسيتايل، ما نريد هذا الديسوشيشن يصير، على مود لا يطلع لي الاسيتايل كوانزيم اي، على مود لا يصير بي انكوربوريشن بمن بالفاتي اسيد سينثيسيس. So will inhibit this step, inhibit the citrate lyase step. شلون inhibit؟ inhibit by this drug hydroxycitrate. In the presence of this الانزيم cannot act on its natural substrate. راح يتيه الستريت الناتشرال 1 اللي هو come or move out from the mitochondria، وراح bind بمن بالartificial اللي هو الهيدروكسيستريتس. So as a result, so the availability of acetyl coenzyme A will be شبيه impaired. So there is no acetyl coenzyme A, so there is no fatty acid synthesis. So this is regarding the medical importance of fatty acid biosynthesis. It's so important and rarely حقيقة actually this is rarely used in our society. Use or indicate or advise our patient, obese patient, to make a regime and to change the lifestyle and do extensive or vigorous exercise to reduce his weight and become a normal or slim person. That's all for today. So glad to answer me in right way. طلاب سجلوا حضوركم لهذه المحاضرة. شكرا جزيلا، وأشوفكم إن شاء الله نلتقي في المحاضرة القادمة إن شاء الله. شكرا جزيلا.